Hello and welcome back to everyone. My name is Shirsha, and in this lecture series we are talking about mammalian cell culture. So if this is your first time visiting my channel, then I would like to brief you up about what we are discussing in this lecture series. So in the previous two lectures we have been talking about mammalian cell culture. In the first lecture, we talked about the various equipments that are required to establish and maintain mammalian cell culture and what is a working principle of each of these equipments and what are their utilities in detail. In the second part, we talked about the various conditions required for cell culture, starting from temperature, pH, gaseous content to media and the different kinds of media that are available, what is the role of the different components of media, so on and so forth. In this particular lecture, we are going to be talking about how you maintain your cell cultures. So this would be the last lecture in this particular series. If this is your first time, then I would suggest that you go back and you watch the previous two lectures to help you better understand what we are going to discuss in this particular lecture. So without further ado, let us delve right into the topic. Maintenance of cell cultures. Now, cultures should be examined daily, observing the morphology, the color of the medium, and the density of the cells. So, the morphology, as you observe it under the microscope, would be indicative of the health of the cells. If your cells are healthy, then they should have a certain specific kind of morphology, depending on what cell type it is. On the other hand, if your cells are not really very healthy, then you could make that out by simply looking at their morphology. If they appear unusually swollen or if their shape is different from their usual shape, then that is typically indicative of deteriorating health of the cells. The color of the media. Now, as we have already discussed in the previous lecture, the media contains certain pH indicators, phenol red to be precise, which gives the media its characteristic reddish color. And as the medium turns more and more acidic, the color of the medium changes from red to bright orange and finally to yellow. So the color of the medium is also indicative of the pH of the medium, which in turn is indicative of what kind of conditions your cells are subjected to and whether your cells are present in a healthy environment or not. And finally, the density of the cells, because you do not want it to be such that your cell density is very high and the amount of medium that you have provided is falling short for that particular density of cells. Or if you're dealing with adherent cells, then you want to ensure that there is enough surface area available for the cells to grow. If the adherent surface is around 90% confluent, then there would be no further space for the cells to grow. And because adherent cells require a flat surface to grow on, as a result, they would tend to grow on top of the other layer, so they will not be growing as a monolayer, and this would cause the cells to simply peel off from the surface on which they were adhered to. So these three things you have to observe daily. So this is where the second glass door of the incubator comes in handy. You can simply open the first door and quickly scan the various cultures kept inside to see if the pH of the medium looks okay. If the pH does not look okay, you take the plates out, you visualize them under the microscope to see, is it because A, that your plates are very confluent, or your cultures are very confluent, or B, is it because there is some contamination, because sometimes contaminations would also cause a change in the pH, or C, is it because that simply there is enough space available on the cell culture or in the cell culture plates for growth, but simply your medium has fallen short and whatever nutrients were there have been utilized and the medium needs to be replenished. So these three things you have to daily monitor without any skipping, because it is very imperative for maintaining healthy cell cultures. So the first topic that we are going to be discussing about is cell growth. Now when cells are seeded into a flask, they enter a lag period which can range from 2 to 24 hours depending on the particular cell type. This is followed by a period of exponential growth, which is known as the log phase, and finally a period of reduced or zero growth after they become confluent, that is the plateau phase. So this is very similar to the growth curve that you will obtain for bacterial cultures as well. You have an initial lag period during which the cells get acclimatized to the new environment, and if you're talking about adherent cells, then they make the initial contacts with the surface essential for survival and further growth. After that, there is a period of exponential growth, and then finally as the plates become confluent, and by confluent I mean that is the area gets occupied, the available surface area gets occupied, the cells enter into the plateau phase. These phases are characteristic for each cell line. So different cell lines will have different lag, log and plateau phase periods and give rise to reproducible measurements. The length of the lag period, the population doubling time in the mid-log phase, and the saturation density at the plateau phase. So all of these characteristics are reproducible given that the environmental conditions, that is the growth conditions, are kept constant. Next we come to feeding. So what do we mean by feeding? Most cells are passaged or at least fed by changing media at least 3 times a week. Again, the frequency of this passaging or feeding will depend on A, your cell type, and B, what kind of containers you're using for growing these cells. If you're using adherent cells, then if your cells are in smaller flasks, then you would need to passage them more often compared to if you were culturing them in bigger flasks. Sometimes your plates will not be confluent or your flasks will not be confluent, but still it might seem that the pH of the media has dropped. Under such situations, you simply need to change the media. You need to replenish the cells with fresh nutrients. So passaging essentially refers to the subculturing of cells. In case of suspension cultures, you can simply add fresh medium into the pre-existing medium. Adherent cultures that do not need to be divided can simply be fed by removing the old media and replacing it with fresh media. So what do I mean by cultures that do not need to divide? For instance, if you're dealing with the terminally differentiated cells which will not undergo further division, in that case, you can simply remove the old media and replace it with fresh media, allowing the cells or providing the cells with nutrients that will ensure their survival. The next thing that we're going to be talking about is subculturing. So passaging is subculturing. Feeding is simply replacing the existing media with fresh media or simply adding some fresh media onto the existing media to replenish the nutrients. Subculturing involves removing the growth media. So if you have your cells in adherent cell culture, then you remove the growth media. You wash the plate to remove any residual trace media. You dissociate the adhered cells. Now this dissociation can be done either enzymatically, and preferentially it is done enzymatically, but it can be done mechanically as well. So enzymatically you can do it by using trypsin. So what is trypsin? It is a protease. It will cleave the cell surface proteins, and in doing so it will cleave those cell surface proteins that are required for attachment of the cells to the cell's culture plate surface. So now your cells would easily detach from the surface. Although some cells may be removed by repeated pipetting or gentle scraping. So this would fall under the purview of mechanical disruption. But keep in mind that mechanical disruption is more harsh for the cells and therefore would not really be preferred because they would affect the health of the cells. So once you have dissociated the cells, then you dilute the cell suspension into fresh media. When the cells become nearly confluent, that is, the culture plates are almost full, several methods are used to remove the cells from the growing surface. So you can either do that by mechanical means, a rubber spatula. So if you go back to the first slide, the cover slide. So over there on the cover picture, what you see is a rubber spatula, which was being used for mechanically disrupting the cells. So you can use a rubber spatula to physically remove the cells from the growth surface. This method is quick and easy, but it is also disruptive to the cells and may result in significant cell death. This method is best suited when you're harvesting many different samples of cells for preparing extracts. That is, when viability is no longer important. When you're at the end stage and you simply want to now collect the cells or harvest the cells for further experimentation, for that situation, mechanical disruption is suitable. However, it is not really advised to go for this mechanical disruption for regular passaging or subculturing of cells. The next method to do so is by using proteolytic enzymes. So trypsin or collagenase are the most commonly used enzymes in combination with EDTA usually. So if you look at the commercially available bottles of trypsin, you will find that it is written on the surface trypsin EDTA. So why? Because EDTA will be chelating the various cations or the divalent cations, which would help in further disruption of the interaction between the adherent proteins on the surface of the cell and the surface of the cell culture plate. So this causes the cells to detach from the growth surface. This method is fast and reliable but can damage the cell surface by digesting the exposed cell surface proteins. Also, you should keep in mind that when you're doing proteolytic digestion or trypsin digestion, you should not really subject the cells to trypsin for prolonged periods of time because this can be detrimental for the cell's health. You subject the cells to trypsin treatment for a very short period of time, typically 1 to 2 minutes is more than enough. Now also the serum that is present in complete media is an inhibitor of trypsin. So that is why after you remove the media, you wash the plate because this washing ensures that any residual media would be removed. So the trypsin once added will not be immediately inactivated. It can serve its purpose, and then to quench the reaction or to stop further or excessive trypsin digestion, you add media containing serum. So the proteolysis reaction can be quickly terminated by the addition of complete media that contains serum. And if you are dealing with suspension cells, then there is no need for either of these methods of disruption. You can simply take the cells, count the cells, and dilute them in fresh medium as per the required density at which the cells have to be maintained. So it's relatively simpler. So the standard procedure for detaching adherent cells is as follows. How do you go about doing it? So the first is medium is drained off or it is removed by pipetting or aspiration, and the cells are washed once with a buffer solution. Typically you wash your cells with PBS, that is your phosphate buffered saline. The cells are then treated with a dissociating agent, most commonly trypsin EDTA. The flask is then incubated until the cells become rounded. So adherent cells, if you look at them under the microscope, when they're adhered, you would see that they are spread out on the surface. And after you treat them with trypsin and then you visualize a cell, you will see that they have become rounded. That is because the interaction between the cells or the attachment points between the cells and the plate surface have been disrupted. So now the cells simply round up and they become loosened when the flask is gently tapped with the side of the hand. So this is actually a good way of seeing whether trypsin digestion is sufficient or not. You add trypsin to your plates. You keep your plates in the incubator because trypsin would function best at 37 degrees centigrade. You keep it for 1 or 2 minutes. You take the plate out of the incubator and you gently tap at its side with your hand. Now, if the cells become loose and they easily detach from the surface upon tapping, then that is indicative that trypsin digestion is complete and you don't need to further digest because that might end up being detrimental. And you can now add complete medium containing serum to quench the reaction. If, however, upon this tapping, you see that the cells are still not coming off, then you should keep it back inside the incubator a little bit longer to allow the cells to be loosened. Because if you don't do that and you simply take out the plates and you add complete medium, so this will cause trypsin to be quenched, further digestion will not take place. And now, because your cells have not completely detached from the surface, you have to do that by mechanically pipetting, and this will be more detrimental for the health of your cells. So next, once trypsin digestion is complete, medium containing serum is added to the plates. The cells are then transferred to a fresh culture tube, centrifuged to remove all of the residual trypsin and collected, or they can be seeded into a fresh flask and diluted as appropriate with fresh medium. So basically, when you have collected your cells after trypsinization, you will count the cells to get an idea of how many cells you have, what is the density, so on and so forth. Then if you want to store the cells, you're going to spin the cells or centrifuge them to form a cell pellet. And then you would resuspend this cell pellet in freezing medium. We will talk about that later, what freezing medium is, what it contains, and so on. If you want to simply expand your cells or subculture the cells, then there are two ways you can do that. You can either do it directly after counting the cells by diluting them as required by you, or you can still centrifuge the cells to obtain the pellet, because this would remove whatever trypsin was present in it. So although the trypsin has been neutralized, but it is still there in the media, so it is advisable that you periodically spin your cells after trypsinization to remove the residual trypsin, and then you count the cells and seed them into fresh plates according to whatever density is desirable. Now, cultures of cells that grow in suspension can be maintained by diluting an aliquot of the suspension into fresh growth medium. So you simply pipe it out a little bit of the already growing cells. You count them, you get an idea of how many cells you need to add to how much medium, depending again on what cell density you want to maintain. And you simply mix that amount of cells along with the media in which it is present into fresh media. So it's relatively much simpler. So next we are going to talk about growth curves and measuring cell growth. So in order to analyze the growth characteristics of a particular cell type or cell line, a growth curve can be established from which one can obtain a population doubling time, a lag time, as well as a saturation density. So if you recall, we just discussed a little while back about the different phases of the growth curve, and then how a growth curve is reproducible for a given cell line given you are maintaining the specific conditions for growth. So now consider that you have a cell line and you're maintaining the required conditions for growth. So you want to monitor how your cells are growing and if they're growing as they should be. So this is where a growth curve comes in handy. So a growth curve will generally show a lag phase, that is the time it takes for the cell to recover from subculture, attach and spread. A log phase in which the cell number begins to increase exponentially. And finally, a plateau phase in which the culture becomes confluent and the growth rate as a result slows down or stops because there is no further space available. However, keep in mind that if you do not passage or subculture your cells when they are in the plateau phase, then it does not mean that they will stop growing completely. They will keep on growing, and if you are dealing with adherent cells, then like as I have already mentioned, these cells will begin to grow on top of the existing cells. So as a result, your cells will begin to detach from the cell culture plates. They would simply peel off from the cell culture plate. So in the log phase, the growth fraction is high and the culture is in its most reproducible form. So it is advised that you passage your cells while they're in the log phase. You don't really allow them to reach the plateau phase. And it is the optimal time for sampling, that is for collecting your samples or for doing any experiments, because the cells are at the peak of their health. An increase in cell number is also a frequently used method of assessing the effect of hormones, nutrients, and so forth on a specific cell type. So, if you remember the previous lecture where we discussed about Ham's nutrient fixture, which was specifically designed to help or to allow the study of the contribution of different components of the medium to cell growth. So if you want to study the contribution of the different constituents of the medium on cell growth, then you would need to have the growth curves, because the growth curve will be representing how well your cells are growing and what is the characteristic of the cell line, like its doubling time and lag time and so on and so forth. So now if you have a growth curve at hand, you can change the amount of the different components you're adding, or you can use different types of, well, let me rephrase. You can either change the concentration of a particular component that you're adding, and you can assess its effect on the cells group, or you can substitute that component for a different component and see, like for instance, you were using a particular source of amino acids. Now you want to try a different source of amino acids. So you can actually see which of these are best suited for your cell's growth and what effect these different sources can have on your cell's growth. So this is what a typical growth curve looks like. So this is your lag phase. Then your cells are getting adjusted to the new medium. Then you have your exponential or log phase. So this growth curve is of HeLa cells in D by F medium supplemented with 5% FBS. Or FBS is your fetal bovine serum. So use of a hemocytometer on an electronic particle counter gives the most direct measurement of cell number and therefore cell growth. So like we were discussing, that you count the number of cells before you passage your cells. So for counting, you use either a hemocytometer where you have to manually count the number of cells, or you can use an electronic particle counter which is automated and therefore will make your life easier. So cells can be counted before, during, and after setting up an experiment to accurately and directly quantitate and standardize the experimental conditions. So this is up to you completely. Depends on what experiments you're doing, what you want to study exactly. So when you will be counting your cells is totally up to you. But for counting of the cells, keep in mind that the cells, if they are adherent in nature, then they have to be detached from the surface of the cell culture dishes to allow for counting. And if you're dealing with suspension cultures, then it actually becomes much simpler. Now the use of a dye such as Trypsin blue when doing hemocytometer count gives the investigator a quantitative standard for the viability of the cells. So when you're counting the cells and you want to know what proportion of your cells are living and what proportion of your cells are dead. So a common way of doing that is using Trypsin blue. So what do you do? Once you have trypsinized your cells, given that they're adherent, or in case of suspension cell cultures, once you have collected the cells, and before you go for counting the cells using the hemocytometer, now keep in mind, because Trypsin blue staining is dependent on visual detection, so you cannot use an electronic particle counter with it. You have to go for manual counting using a hemocytometer. So once you have your cells to be counted, let's say in a microcentrifuge tube, you mix it with Trypsin blue, you incubate it for a short period of time, and now you load these cells onto your hemocytometer and visualize it under the microscope. So living cells will exclude the dye. So cells that have excluded them would be considered to be viable, whereas dead cells would take up the dye. So by now counting the number of cells that have taken up or excluded the dye, you can obtain a differential count of the living versus dead cells. So this is what a hemocytometer typically looks like. So these are the glass slides. This is commercially available. And in this particular area, you have these grids prepared on which will actually help you to count your cells. So these grids provide an outline or a layout, or rather a boundary within which you have to count your cells. And then there are specific calculations available. I'm not really going to get into the details of that, by which you can obtain the density of cells present in your starting culture from where you loaded the sample onto the hemocytometer. So this is a view of the hemocytometer under the microscope. See, you can see these grids. So each of these big grids have smaller grids within them. And you can see the cells over here rounded because these cells have been trypsinized. So the live cells are white. They have not taken up any stain. So if we are going to mark out the alive cells, then this is your alive cell, this is your alive cell, and this is your alive cell. And the rest of the cells, they have taken up the blue dye, so therefore they are dead. So staining with trypan blue will give you an idea about the status or the health status of your cells. Cell viability can also be measured by an Mtt assay. So what is that? Yellow MTT is a tetrazole. So MTT, the starting substrate, is yellow in color, and this is reduced to purple pharmazine in the mitochondria of living cells. So if you have living cells, then if you add yellow MTT, you're going to get purple pharmazine. And if you have dead cells, then this yellow compound will remain unchanged. Why? Because only the mitochondria of living cells will catalyze the conversion of MTT to pharmazine. The absorbance of this colored solution can be then quantified by measuring at a wavelength between 500 and 600 nanometers by a spectrophotometer. This reduction takes place only when mitochondrial reductase enzymes are active, and the mitochondrial reductase enzyme would be active only in, as you can guess, the living cells. So therefore, the conversion can be directly related to the number of viable or living cells. Greater the number of viable or living cells, greater would be the conversion of yellow tetrazole to purple pharmazine. So this is what it looks like. So if you go along the row, you can see that the intensity of the purple color is decreasing. So this is indicative of lesser and lesser amount of living cells. So if you simply dilute your cells, then as you go down the dilution, this is the most dilute on the right hand side, and on the left hand side you have the least dilute, that is the highest number of cells. So the intensity of this purple colored product formed is differing. So increasing amounts of live cells will result in an increase in purple color. So this will act as a visual measure of how many live cells you have. So in the previous case where you were using trypan blue, the count that you were obtaining was a differential count, because there you could exactly count the number of living cells and the number of dead cells in a given population. But over here, you will not really obtain a direct count of the living cells present, but rather it is more of an idea of what proportion of cells can be living. But you will never really know, or rather it won't really even give you an idea of what proportion of cells are living. It will simply tell you how, whether you have more living cells or less living cells. It will never give you an idea of the proportion of living to dead cells. So for that, trypan blue is better. So next we are going to talk about contamination. So contamination is one of the most difficult things that any person working with cell culture has to deal with. It is something that everybody wants to avoid, but they know that no matter how hard they want to avoid it, it is unavoidable. No matter how careful you are, somehow it will creep in. If you're lucky, then you will not have to deal with it very frequently, maybe once in every 1 or 2 years, but yes, it will be there. So if you do practice good cell culture practices, and if you follow all protocols and take care of sterile conditions and everything, then you should be able to avoid contamination, theoretically speaking. But being a researcher who works in the subcontinent of India, where the humidity is very, very high, so certain times of the year, like monsoons, you have to be even more careful than usual because over there the risk of contamination is greater. So, this is one word that can raise fear in the hearts of any person working with cell culture. So one occurrence that every person who tries to grow animal cells in vitro has to deal with sooner or later is contamination. If you're lucky, you have to deal with it less frequently, and I hope that you are lucky. Now, as a problem, it can vary from irritating to catastrophic. Bacterial, mold, and fungal contaminants are the most common because they are designed by nature to be resistant to desiccation and to remain for long periods of time in a dormant state, but to immediately replicate when they find a good source of food, such as cell culture media. And bacteria is one of the most pesky things that you can think about. It is present everywhere, they are ubiquitous, they are so difficult to get rid of. Because of their ubiquity, size, and fast growth rates, bacteria are the most commonly encountered biological contaminants in cell culture. Bacterial contamination is easily detected by visual inspection of the culture because your infected cultures will appear cloudy, that is turbid. So typically if you are growing cells in, let's say, Petri plates, and you take up the Petri plate, it's just slightly tilted, you can easily see through the media. It's crystal clear. But if you have any bacterial contamination, then your media will turn turbid immediately. So simply by looking at the plates, you can say that yes, you have bacterial contamination. And sometimes there can be even the formation of a thin film on the surface, depending on how intense your contamination is. Also, if you have bacterial contamination, the pH of the media changes. So your plates will now take on a yellow coloration. That is also indicative of contamination. Sudden drops in the pH of the culture media is also frequently encountered. Under a low power microscope, the bacteria appear as tiny moving granules between the cells, and if you observe them under a high power microscope, then you can even resolve the shapes of the individual bacteria. So it is very easy to visualize bacteria under the microscope. So if you're observing at low power, let's say at 10x focus, then you would see like these dot granulation in between your cells. You will see this intensive granulation between your cells, or which we sometimes refer to as a background. So this is indicative of bacterial contamination. And if you focus it at high power, let's say at 75x or 100x, you can easily make out the shape of the bacteria. And not only that, and I would say this from personal experience, you can even see the bacterial cells as they're moving and wiggling their way, making their way through the medium, and how they're attaching to your cells. You can see everything. Well, you can't really see them attaching, but you can definitely see them wiggle through the media and go and hit your cell. So like bacterial contamination, cultures contaminated with yeast also become turbid, especially if the contamination is in an advanced stage. So getting yeast contamination is pretty easy. Let's say you had some toast in the morning and you just went in to do cell culture, and then you get the crumbles of toast from your t-shirt onto your cell culture medium. So that can be a very common source of yeast contamination. And that is why, to avoid contamination, we typically take a lot of precautions. So mostly you would see that your cell culture facilities have a double door system. That is, you first open the first door which leads to outside, and you enter into this secluded space. Once you've closed the first door, then you open the second door, and now you enter into your cell culture room. Also, your doors mostly would be fitted with one of those things that dispense air at you at a high pressure when you're entering the door. So this will typically remove any spores of yeast or bacteria or these toast crumbs that you might have on your t-shirt or on your clothes. It is always advisable to wear lab coats when handling mammalian cell culture or any cell culture, because this will provide an additional protective layer and anything on your clothes can't really fall onto your work surface. And of course, you'll be wearing gloves when working with your cells, and you sanitize or sterilize your hands periodically with 70% ethanol. You sterilize everything that you're going to work with. So precaution is better than prevention. I'm sorry, prevention is better than cure. So there is very little initial change in the pH of the culture that is contaminated by yeast until the contamination becomes heavy. So yeast contamination is more difficult to detect than bacterial contamination, and likewise it is more difficult to get rid of than bacterial contamination. Under the microscope, yeast appear as individual ovoid or spherical particles that may bud off and give rise to even smaller particles. So it depends on what stage you are observing. So you can have either individual ovoid or spherical particles, or you might have some particles from which younger yeast are budding off. You can see that as well. So this is just an example of a bacterial contamination being viewed under high power microscope. So over here you have your cells which are being viewed by a phase contrast microscope, and then you have these rod-shaped bacteria, the bacilli, which are present just everywhere. And this is a visualization of yeast contamination again under high power microscope. So see, you can see these yeast particles as the spherical, some are spherical, some are ovoid. Some cases you can see there's a small bud coming off from the bigger one. In this particular one, let me point it out. So in this one you can see the bud coming out from the bigger sphere. So depending on what stage you're actually visualizing, you can see it. Now mycoplasma, they grow quite slowly compared to bacteria and yeast, and do not immediately or directly destroy the cells. So these will not lead to direct death of your cells, but rather they would alter the functionality of your cells. So this will leave you even more befuddled that what's wrong? I don't see any visual contamination and still my results are not working. So what is happening? However, they are known to alter the function and metabolism of the cell culture and interfere with nucleic acid synthesis. So this is even more dangerous than the previous one. Moles are eukaryotic microorganisms in the kingdom of fungi that grow as multicellular filaments called hyphae. A connected network of these hyphae are referred to as a colony or a mycelium. Now, similar to yeast contamination, the pH of the culture remains stable in the initial stages of mold in contamination, and then it often increases or decreases as the culture becomes more heavily infected and becomes turbid. Under microscopy, the mycelia usually appear as thin wisp-like filaments, and sometimes you can even visualize them as denser clumps of spores. Again, this would depend on what reproductive stage of the mold you're actually visualizing under the microscope. Spores of many mold species can survive extremely harsh and inhospitable environments in their dormant stage, only to become activated when they encounter suitable growing conditions. And this is the problem with all contaminants. You might think that they're dead, but no, they're just dormant, and they get the appropriate condition and bam, they begin to grow. So this is a visualization of fungal contamination under the microscope. See, you can see these big clusters of spores over here. Yeah, so these are your cells under the phase contrast microscope, and these are your clusters of spores. We also see some specks like you would typically see in bacterial contamination. This is how turbid your media turns when it is contaminated. So you see, if you can recall the previous images of media that we have seen, then they would typically be crystal clear, translucent, and over here it is completely opaque, it is completely cloudy, and you see the color of the media, it has also changed. We had a bright reddish orange color, and over here it is completely yellow, which indicates the fall in pH. So yeah, this you can simply look at it and say that your culture is contaminated, but this is not always the case. Typically with bacteria, this is what you get. With fungi, only in advanced stages of contamination will it begin to look like this. So it is more difficult to detect. So this is again where the importance of daily visual inspection of your cells come in, because if there is some yeast or fungal contamination, then you should be able to visualize it as some background on your plates when you're observing it under high power microscopy. It won't be really evident by simply looking at the culture plates, but under the microscope, if you have a trained eye and if you know what you're looking for, you can detect fungal contamination at earlier stages. So next we are going to talk about the different types of cell culture. Now that was enough talk about contamination, and that was enough negative talk. Well, precautionary talk, not really negative talk. And now we're going to talk about the different types of cell culture. So several different types of cultures are routinely performed in laboratories. These can be divided into primary cultures and the culture of established cell lines. Now this is something that we have very briefly touched upon in the first part of this lecture series on mammalian cell culture, and we are going to discuss it in more detail now. So most mammalian cells in culture can be divided into three basic categories based on their morphology. So before we get into the distinction between primary and established cell line culture, we should talk about the three categories or three basic categories into which mammalian cells can be divided based on their morphology. So we have the fibroblastic or fibroblast-like cells, which are bipolar or multipolar in nature and have elongated shapes. They grow attached to a substrate. So these fibroblastic cells are typically adherent cells. So this is what they look like when visualized under the phase contrast microscope. So if you recall the contamination images we were just seeing, those were of fibroblastic cells. You see, they have this typical spindle shape characteristic of fibroblasts, and hence the name. So the next type of cells that we are going to talk about are the epithelial cell-like cells. Epithelial-like cells are polygonal in shape with more regular dimensions, and they grow attached to a substrate. So this is what your epithelial-like cells would look under the phase contrast microscope. So as you can see, they are more regular compared to the fibroblastic cells, and they are also adherent in nature. And finally, we have the lymphoblast-like cells or the lymphoblastic cells. These are spherical in shape and usually grown in suspension without attaching to a surface. So this is what your lymphoblast-like cells look under the phase contrast microscope. So you see, adherent cells have a distinct characteristic shape, either spindle-like or polygonal, whereas the suspension cells are more rounded. Why? This is because the adhesion actually causes these shapes. And in suspension, because there is no attachment, so no shape is seen as such. They just all appear rounded. And this is what your cells would look like your fibroblastic or epithelial cells would look like this once you trypsinize them. So something that I accidentally skipped forward in the previous slide. In addition to these three types of cells that we have discussed, there can be other cellular shapes as well for certain specific cells. So for instance, if you are dealing with neurons, then you can't really classify them into either of these three cell types based on their morphology, and they have their own distinctive morphology. So next we are going to talk about adherent versus suspension cell culture. Now there are two basic systems for growing cells in culture, either as monolayers on an artificial substrate, that is adherent culture. Now keep in mind the key word over here is monolayer. These adherent cells grow best when grown in a single layer, and if the cells pile up on top of the other, then this would simply lead to their detachment, as I have repeated so many times already throughout this particular lecture. Or you have the free floating cells in the culture media, that is the suspension culture. The majority of cells derived from vertebrates, with the exception of the hematopoietic cell lines and a few others, are anchorage dependent. Why? Because think of how these cells reside within the body itself. All of these cells are embedded in an extracellular matrix. They're not really freely flowing within the body system either. So therefore, most of these cells are anchorage dependent. And this anchorage is essential not only for their proper survival, but also for their optimal functionality. And these cells therefore have to be cultured on a suitable substrate that is specifically treated to allow cell adhesion and spreading. That is, you use tissue culture treated either glassware or plasticware for this particular purpose. And these artificially provided surface has to be inert and it has to be non-toxic. It should not be in any way detrimental to your cells and it should in no way affect the growth of your cells. It should be totally inert. So if we are to look at a table of differences between the two types of cell culture, so adherent cell culture is appropriate for most cell types, whereas suspension cell culture is appropriate only for cells that are non-adhesive in nature. Adherent cell culture requires periodic passaging or subculturing, but allows easy visual inspection under the inverted microscope. You can simply take your glassware or plasticware, which can be either a petri dish or it can be these multi-well plates or it can be a flask, like those flattened flasks that we were seeing in images so far. And you can simply take it, put it under the microscope and visualize the cells. In contrast, for suspension cell cultures, it is easier to passage because no trypsinization is involved. You simply count the cells and supplement them with fresh media or you dilute them in fresh media. But it requires cell counts and viability determination. In suspension culture, typically you would go for viability determination by trypan blue staining, which you would generally not do so commonly for adherent cultures. And it requires cell counting. And if you want to visualize your cells, you can't just directly take the cell culture flask and put it under the microscope and see it because you don't really have a single layer of cells. So in this situation, you always have to use a hemocytometer for visualizing your cells. Culture can be diluted to stimulate growth because if you dilute it or when you dilute it, you supplement it with fresh nutrients and this can stimulate growth. Adherent cells are dissociated either enzymatically by using trypsin or mechanically, and we have already discussed about the pros and cons of both of these mechanisms. Suspension cells do not require either any enzymatic or mechanical dissociation. You can simply just dilute it into fresh media. Now for adherent cells, the growth would be limited by the surface area and typically you split or subculture the cells when your flasks or when your plates are at 70 to 80% confluency. Whereas in suspension cells, growth is limited by the concentration of cells in the media. If you allow it to go beyond a certain point, then your proportion of dead cells would keep on increasing. Adherent cell culture requires specific vessels which are tissue culture treated, whereas for suspension culture, you can maintain it in culture vessels that are not tissue culture treated, but it requires constant agitation for adequate gas exchange. If you do not subject it to constant agitation, then your cells are going to settle down and that is again not desirable because these cells, as the name suggests, should remain in suspension. So now we're going to talk about primary culture in a little bit more detail. A primary culture consists of a culture started from cells, tissues or organs taken directly from the organism. So in primary culture, you need to have an organism which will be your donor organism. From there you take the cells and tissues and you start the cell culture. The tissue is disaggregated or separated by enzymatic or mechanical methods. Again, I would say that enzymatic methods are preferred over the mechanical ones because of their less harshness. The cells that survive this disaggregation process will attach to the cell cultured vessel or survive in suspension depending upon its characteristic nature and proliferate to form a primary culture. Now, these cultures can contain mixed cell types because it is quite natural when you're isolating cells or tissues from a whole organism, it will be difficult to isolate a single cell type. So, what you will be isolating is a mixture of population of different cell types, or it might consist predominantly of a single cell type. But however, in this scenario also there are some other cell types, maybe much fewer in number compared to your predominant cell type. However, given the complex interrelationships of cells in any organ or tissue, primary cultures seldom, if ever, will consist exclusively of a single cell type. It is really unlikely. They are usually heterogeneous and have a low growth fraction, but are more representative of the cell types in the tissue from which they are derived and in the expansion of tissue specific properties. So obviously they will be heterogeneous because it will be a mixed population comprising of different cell types, but they are a better representation of the specific tissue from which these cultures have been derived, and as they expand in culture, they will retain these tissue specific properties, like for instance the relative proportion of the different cell types in that tissue. Now, attempts may be made to mechanically or enzymatically purify the cell type of interest during tissue dissociation. So you can do that. During the dissociation process, you try to purify your cell type of interest, or by culturing the cells in conditions that specifically kill or inhibit some major contaminant cell types. Contaminant in the sense that it is an undesired cell type. You do not want it in your culture. Alternatively, primary cultures can be maintained in conditions chosen to positively select for the survival of only one cell type. So you then either go around trying to eliminate the other undesired cell types either mechanically or by imposing certain conditions that are detrimental to them, or you can go about it in a positive manner where you're not really trying to kill the cells that are undesired, but rather you provide conditions in which only your cells of interest will survive. So in this way also these other cells will die, but your aim is to promote better survival of your cells of interest, not killing the cells of disinterest or not non-interest. In general, the method chosen must take into consideration the desired balance between obtaining a high cell yield and minimizing damage to the cells to be cultured. So, irrespective of what method you are selecting, you should ensure that your method of choice is best suited for the survival of your cells of interest and it minimizes the damage to these cells. Considerations of the earliest time the cells are to be studied after recovery and the maximum time that they are to be kept in primary culture might also determine the isolation method of choice. So it will also depend on what kind of study you want to do, at what stage you want to study the cells after primary culture establishment. While the majority of cell culture studies use established cell lines, there are some instances in which primary culture is preferred. So again, this is very study specific. Study of normal terminally differentiated cell types like neurons or T cells in vitro would require primary culture since these cell types do not divide in vivo. And for studying these terminally differentiated cells, you don't really even get a chance to replicate these cells because they are no, I'm sorry. I take that back. So, yeah, if you're studying with neurons, say for instance, not for T cells, but for neurons, they don't even replicate further. They will not even divide. So, you establish the cells, you do your study, and that is the end of it. You can't really maintain them. While others, like for example, let's say smooth muscle cells that you have isolated from the lungs of your subjects, that you can maintain for only a certain number of cultures, a certain number of passages, but definitely not for very prolonged periods of time. In addition, it has proved difficult with some dividing cells to fully maintain their differentiated state in vitro through multiple passages. Under that situation also, you can only maintain them for a certain number of passages, and after that you have to go back to the beginning and establish the cell culture again. So this makes it actually more difficult to deal with. Finite cell cultures are formed after subculturing of a primary cell culture because, as we have already discussed in our previous lectures on this topic, each and every living cell can only divide a certain number of times, after which it stops dividing. So these primary cell cultures would essentially comprise of finite cell cultures. However, these continuous cell cultures or the commercially available established cell lines, they have been transformed either by some chemical means or by using some viruses, as a result of which they can proliferate indefinitely and they are therefore immortalized. Now, these finite cell cultures or primary cell cultures, because they are finite in nature, will proliferate for only a limited number of cell cycles, after which they will undergo senescence and they will die. Proliferation of such cells in vitro are related to the degree of differentiation. In general, cultures that are derived from embryonic tissues, they will divide more, they will survive longer, they will proliferate better compared to those derived from adult tissues. So it is simply representative of how those cells would survive when normally present in the body. Normally in the body, embryonic cells would divide more, survive more. So will they in cell culture. Normally in the body, adult tissue cells will not divide so much. So they would not divide so much in primary cell culture either. This presumably reflects the lower level of specialization and presence of a greater number of replicating precursor or stem cells in the embryo. And that is why embryonic primary cell cultures will survive longer because they have more number of stem cells. Adult tissues, on the other hand, usually have a very high proportion of non-replicating specialized cells, often within a more structured ECM or extracellular matrix. So therefore, propagation is more difficult and the lifespan of the culture is often shorter. Now, how do you go about setting up a primary culture? So you have your host organism from where you know your tissue of choice or interest that you want to take. So this tissue is cut into small pieces until there are no discrete tissue pieces observable with the naked eye. So you chop up the tissue into tiny pieces till you can't really make out any large chunk or piece with the naked eye. Now you can either do it mechanically or the tissue can be dispersed enzymatically as well by trypsinization. And like always, this enzymatic means would yield better results compared to mechanical disruption. Now, once you have disaggregated your cells, the cell suspension is carefully pipetted into a centrifuge tube. The cells are washed repeatedly by centrifugation and careful resuspension. So what do I mean by that? So you take your cells, you collect it in a centrifuge tube, you spin it to pellet down the cells. You remove whatever supernatant is there, you add certain buffer to it, and you resuspend the cell pellet in the buffer, centrifuge it again, get the pellet, discard the supernatant. Add the washing buffer again and you repeat this process a couple of times to wash the cells and to remove any traces of the extracellular matrix and other enzymes and other fluids that were already present in your tissue from where you were isolating the cells. The cell pellet is then finally resuspended in the required media and added to the cell culture dishes. The plates are kept at 37 degrees centigrade if it is a vertebrate or if it's a human cell. Well, not human. Let's say if it's a vertebrate, can be human as well. In a 3 to 5% CO2 humidified incubator. Like we have already discussed previously, humidity is also very important for optimal cell survival and growth. The plates are regularly observed under an inverted microscope to help for regular monitoring, to allow regular monitoring and maintained as necessary. So let us take an example where you're creating, let's say, a cell line from the ear of this mouse. So or let's say maybe, from the lungs. let's not go with the ear, the lungs, because that's more um, something that you would usually do. So you dissect the mouse, you take out its lungs, you cut it up, you chop it up first physically, and then you add enzymes to help for enzymatic disaggregation. So now you have your cells collected in the centrifuge tube, and you have spun it repeatedly, and you have washed the pellet. Now you have resuspended it in your medium of choice, and you grow it in these flasks. Once your flask is confluent, then you subculture it or passage it, and you simply expand it as required. Now coming to cloning of these cultured cells. So these cultured cells, let me remind you, are not clones, because a clone would be a situation where all the cells are derived from one single starting cell, but over here you have a lot of starting cells from which the culture is derived. So now how do you clone or generate a clone from these cells? So due to the heterogeneous nature of animal organs, primary cultures potentially contain more than one cell type. As interpretation of data arising from study of such mixed cell populations can be difficult, cell lines are often cloned. Because yes, it would be more difficult to interpret the data if you have different cell populations. Then you can't really say what effect you're observing because of which cell population. So for that, cloning becomes imperative or essential. Cloning a cell means deriving a population of cells from a single cell. So how can you do it? The first way of doing it is by dilution technique. So in this, what you do, your cells are resuspended in growth medium at a known concentration. The concentration is then adjusted to 5 to 10 cells per ml, and aliquots are added to each well of a microtiter plate. And you then check visually to verify which of these wells contain a single cell. So you dilute it such that you have only 5 to 10 cells per ml, and now you aliquot it into the microtiter plates. And then you have to visually inspect each of these wells to see which of them has a single cell. Because let's say if you have 5 cells per ml and you're pipetting 250 microliters or 500 microliters. So if you have 5, then okay. So if you have 5 cells per ml, then that means you have 1 cell per 250 per 200. I'm sorry, per 200 microliters you have 1 cell. So you are pipetting 200 microliters into each well of a microtiter plate. And now you'll be visually inspecting each of these wells to see which well has a single cell. Now you allow these cells to grow in the microtiter plates itself. Following growth of the clones to approximately 50% confluency, that is when 50% of the surface area is occupied, the cells are passaged and then cultured in increasingly larger wells and flasks. Now you can't obviously take the cells directly from one well of a microtiter plate to a big culture dish, because then the cell will be overwhelmed in such huge space with so much media. So you have to gradually increase the cells, right? And also if you have it at like 5 cells per ml, and then you will have 1 cell per 200 microliter, because 1 ml has 5 cells. So 5 cells in 1000 microliter and 1 cell in 1000 by 5, 200 microliters. So 200 and 250 microliters is approximately the volume that you can pipe it into a single well of a microtiter plate without risking any overflowing. And you would obviously not want to fill up your microtiter plate up to its brim. So like these are the wells of the plate, right? You will not want to fill up the plate right up to the brim. So you would typically pipe it 100 to 200 microliters to leave some space on top. So this is an example of cloning of adherent cells using the dilution technique. And over here, the cells have been fixed and stained using 1% crystal violet. So this allows your cells to be visualized. So you see this particular well has 1 cell, this is 1 cell, this is 1 cell. So obviously you're not going to fix and stain your cells, because that will kill your cells. You will identify which of these wells have a single cell by visually inspecting each well under the microscope. And once you've identified it, you will let it grow, and when it is at 50% confluency, you will subculture it. The next technique of doing this is by using a cloning rings. Now, what do you do here? So after plating adherent cells at a density of 400 to 600 cells per 15 centimeter culture dish. Again, this is just a rough approximation and you can change it as per your requirement. The individual clones are allowed to grow till they are several millimeters in diameter. The location of the clones are to be removed are then marked. So because each clone is a group of cells derived from a single cell, so you mark the location of the clones which you are to remove. Sterile polystyrene or steel cloning rings which have been dipped in grease are placed over individual colonies. The medium is then removed and a small amount of trypsin is added. So you plate the cells in such a fashion that you have individual cells at different distances apart and you let them grow. So you have now colonies which are essentially clones derived from the single individual cell. Very similar to plating in case of bacterial cell culture, where you want to obtain independent clones of cells or colonies which are independent clones essentially. Now over here, because in order to collect the cells, you have to trypsinize them because these are adherent cells. Now if you trypsinize them, then all of these cell population will simply end up getting mixed up. So to avoid that, these polystyrene or steel cloning rings are available. You dip them in grease to ensure that they're tightly sealed onto the cell culture plate. And when you add trypsin, because the cells would be in a liquid medium, they don't just simply slip out from underneath the ring. If there is a grease, that will seal the ring in position. And now when you trypsinize the cells will remain within the ring and you can just collect it now. After the cells have detached, the trypsin is diluted with medium containing FBS. Clone cells are then collected from inside the ring and transferred first to culture wells and then to increasingly larger culture vessels for further work. So just like in case of dilution technique, once you have collected the cells, you grow them in increasingly larger culture dishes. And FBS is your serum. So when you add serum to trypsin, as we have already discussed, that will terminate the trypsinization process. So this is what cloning rings look like. So you have your culture dish plate and you have these plastic uh, cloning rings. Obviously they have to be sterile before use, and you just apply grease to the bottom surface, fit it on the plate, and then the cell which is within it, you trypsinize it and you collect it from within by using a pipette. So next we are going to talk about the culture of established cell lines. So we have already mentioned established cell lines in the first lecture of this particular series. So a little bit more discussion on the same. The second type of cell culture is the culture of established cell lines. Continuous cell lines. Now these established cell lines, they are typically immortalized because these are derived from either cells that have been transformed within the body itself before they were derivatized to establish a cell line, or some cells that have been transformed either chemically or by using some viruses to ensure that they're immortal and that they can divide indefinitely in nature. So that is what makes them continuous. Continuous cell lines involve cells that proliferate almost indefinitely in culture, provided you provide them with appropriate medium and appropriate space. The vast majority of these are derived from tumors, so these were already transformed before they were derivatized to give rise to cell lines, or cells that are transformed in vitro, although some of the very earliest lines were established from normal embryonic tissue. For example, the 3T3 mouse embryo fibroblast. Now, transformed cell lines usually have an increased growth rate and higher plating efficiency as compared to primary cell cultures. They are continuous in nature, require less serum in media because they are more adapted to growing in cell culture. But they have undergone a permanent change in their phenotype through some genetic transformations, which are either chemically induced or virally induced. These cell lines, after many decades of growth in laboratories, are also relatively tough. That is, they are resistant to temporary lapses in good cell culture technique. So with primary cell cultures, you have to be much more careful and much more vigilant. With continuous cell lines, although you can afford to not be so vigilant, but it is advisable to not do so, and it is advisable that you maintain proper cell culture practices and take good care of your cells if you want the best results. In 1951, the first immortal human cell line, HeLa, was established, and I'm pretty sure everyone has heard of it. The cell line was derived from cervical cancer cells taken from an involuntary donor, Henrietta Lacks, who died of cancer later that year. The cells were propagated by George Otto Gay without Lax's knowledge or permission, and later on it was commercialized, and then a subsidiary was paid to the family members of Henrietta Lacks. So HeLa, Henrietta Lacks. That is what it stands for. Lastly, we are going to talk about cryopreservation. So by now we already know how important it is to maintain your cells in a healthy and good fashion. But however, no matter how hard you try, contaminations may be unavoidable. So in case you have contaminations, then you have to throw away your cells and you have to restart the culture. Or in case you have been culturing the cells for a really long period of time, and although theoretically speaking these cells are immortal in nature, the continuous cell lines, but sometimes their functionality might get affected after a lot of passages, and then your experiments might not work properly. Not all, but some experiments. So under such situations, you need to go back to cells of earlier passage. Now, how do you store these cells? So for that, you have cryopreservation. Now, cell lines in continuous culture are also subject to genetic drift. They're susceptible to microbial contamination, and finite cell lines will simply undergo senescence. As an established cell line is a valuable resource and its replacement is expensive and time consuming, it is extremely important that they are cryopreserved for long-term storage. The best method for cryopreserving cultured cells is storing them in liquid nitrogen at a temperature of minus 196 degrees centigrade in complete medium in the presence of a cryoprotective agent like DMSO or dimethyl sulfoxide. Now, what is the utility of this cryoprotective agent? The cryoprotective agents will reduce the freezing point of the medium and allow a slower cooling rate, therefore greatly reducing the risk of ice crystal formation, which would otherwise damage the cells and cause cell death. Most cell cultures can be cryopreserved in a mixture of 80% growth medium supplemented with 10% FBS and 10% DMSO. So addition of serum is also important to the media for cryopreservation, and sometimes for certain cell lines your cryopreservation media comprises of entirely serum supplemented with your cryoprotective agent DMSO. So this is what these cryopreservative vials look like. And if you recall, one of the essential equipments for establishing cell culture were liquid nitrogen tanks to allow for storage of these cryopreserved cells. So this brings us to the end of our lecture series on animal cell culture or mammalian cell culture. So to briefly recapitulate, this was a three-part lecture series. In the first part, we talked about the different equipments commonly required for cell culture and their principles, how they act, and what are their utilities. In the second part, we talked about the different conditions for cell culturing and the different requirements like temperature, pH, media, serum, humidity, gaseous content, so on and so forth. What are the utilities of each of these components, what effect they have on your cell culture, so on. And finally, in the third lecture, that is in this particular lecture, we talked about maintenance of cells, how you maintain them. We talked about adherent cells and suspension cells and some critical differences between the two. We also talked about primary cell cultures and how you can establish them. What are their advantages, the pros and cons. We also talked about continuous cell lines, and finally, last but not the least, we talked about cryopreservation. So this brings us to the end of this vast topic. I have tried to describe it to the best of my abilities. In case you have any doubts or any questions, please feel free to leave it in the comment section and I will surely get back to you. If you like this video, then do share it and subscribe to my channel for more such content. So that's it for this time, and I'll see you all again next time. Bye.